Okay, this video is about ketogenesis, and ketogenesis is going to be the production of ketone bodies, which is going to be in response to low glucose levels in the blood, and it also makes ketone. Ketogenesis occurs when you have you use up all your glycogen storage, so you can produce ketone bodies this way. And ketogenesis occurs in humans. It's going to occur in the mitochondria of liver cells in your liver. So first of all, if we're going to start off with beta, it's basically you're going to be breaking down fatty acids to get acetyl-CoA, and this is acetyl-CoA. It's just 2 carbons with the enzyme coenzyme A attached to it. This is 2 moles of it up here. This is what you start out with, and this is you get acetyl-CoA when you break down fatty acids by beta oxidation. So start of ketogenesis. We're going to start off with 2 moles of acetyl-CoA. First enzyme is that it occurs is going to be it's called thiolase. And what thiolase is going to do, it's going to be a condensation reaction, and you're going to have one of these coenzyme A's are going to leave, and when they leave, you're going to form a carbon-carbon bond between these two right here. It's going to connect them. So one coenzyme A leaves, and you form a carbon-carbon bond, and that's going to produce acetoacetyl-CoA. And next, acetoacetyl-CoA, you're going to have the enzyme HMG-CoA synthase, and that's going to produce HMG-CoA. And how it does that, it's basically, it adds another acetyl group. Another acetyl-CoA comes in, its coenzyme A gets cleaved, and it gets added onto acetoacetyl-CoA, which gives you this long word, which is beta-hydroxy beta-methyl glutaryl-CoA, or for short, just HMG-CoA, which you could see both on the test. So I would recommend learning both names. And then HMG-CoA is also a precursor for cholesterol synthesis. So from right here, this is where you can make cholesterol too. But if we keep going throughout the ketogenesis cycle, we're going to see you run into HMG-CoA lyase next, and what the lyase does, it's just going to cleave an entire acetyl-CoA. It's going to cleave one of these off, because this has an acetyl-CoA attached at the end. It's going to cleave the whole acetyl-CoA off, and you're just going to be left with your first ketone body, which is going to be acetoacetate. That's going to produce acetoacetate. And I would definitely know all the enzymes and what they do step by step. Remember that, this is occurring in the liver. I would know the structures, just to being safe, I would know the structures of acetyl-CoA, which is really easy. And I would definitely just know the structure of acetoacetate. I wouldn't worry about these other structures because they're really complex and you're not going to see them most likely. But acetoacetate you definitely see. And once you get to this step, a couple things can happen. And that's going to be part 2 of this video. So if you just click on part 2, and we'll continue from there. Okay, this is part 2 of ketogenesis. Where we left off is we just formed acetoacetate. I would know the structure of acetoacetate also. Okay, so once we get to this part, we have a couple different ways we can go. Acetoacetate, some of it can get degraded to acetone, which is the second ketone body. So this is the first ketone body. Acetone's the second ketone body acetoacetate is going to form, and that it forms acetone by the enzyme acetoacetate decarboxylase. It's just going to remove this carboxyl group from acetoacetate, giving you acetone. Acetone's a waste product. It's just going to get excreted in your urine. Another route we can go is by using NADH+, that coenzyme, plus the enzyme D-beta-hydroxybutyrate dehydrogenase is going to form this product, which is called beta-hydroxybutyrate, which is the third ketone body that forms from acetoacetate. So you have acetoacetate, the first ketone body, and from acetoacetate it can break down and form 2 other ketone bodies, which is going to be acetone and beta-hydroxybutyrate. Okay, I would know the structure of beta-hydroxybutyrate also, just to be sure. And then the last pathway we can go is acetoacetate just straight up enters the bloodstream. And beta-hydroxybutyrate also will enter the bloodstream. So once in the bloodstream, these 2 ketone bodies can get broken back down to acetyl-CoA, which then can go back into the TCA cycle, which can be used to fuel energy for muscle or brain. And for your brain, the beta-hydroxybutyrate is the main, can be your main energy source for your brain when you have low blood glucose levels. And that's really the take-home whole like picture is what you need to know about ketogenesis. I really just memorize all the enzymes, what they do. Just go take it slow, go step by step. Just memorize the structures of acetone, beta hydroxybutyrate, and acetoacetate, and know that those are the ketone bodies formed, and know that you can get energy once it's in the blood. That's something you can break. It can get rebroken down into acetyl-CoA, which then could be used in the TCA cycle to generate ATP. One more thing is once acetoacetate and beta hydroxybutyrate get into the blood, these ketone bodies are very acidic and can a lot of them can drastically lower the blood pH. By lowering the blood pH is a really bad thing. A lot of bad things can happen once you start lowering your blood's pH. So that would result in what's called ketoacidosis, or ketosis is another name for it, and that's not too good when that happens. But the whole thing is that's the whole thing you need to know. Just memorize the enzymes and their structures, and you should do very well on this section of the test.